Sven, it's a great pleasure to welcome you at the Collaborative Research Center, Origin and Function of Metaorganisms. Very nice that you made it the way down from Stockholm to sunny Kiel. I'm very pleased since I see in front of me one of the fathers of the gut-brain microbiome axis. And there's, for me personally, a very influential paper, which is the Heitz et al. Paper in 2011 in PNAS, which long before I knew you, I used in lectures, in public lectures, in particular, the figure with the in situ hybridization of the NGF gene or something like that. Yeah. That was a landmark paper at that time. Now it's the gut-brain axis field. How did you get to that, discovering that there is a gut microbiome brain behavior influence?
It goes back actually to my early days in Singapore, because I started in Singapore to support the Karolinska Institutet to build relations between KI and the growing activity in biomedical research in Singapore. And there was a guy at the Genome Institute, his name is Ed Liu, and he basically said, I help you to do expression arrays. So we did expression arrays of an entire animal not exposed to living microbes, the germ-free mice. And we ran that compared to a mouse exposed to microbes. And we still have a lot of unpublished data from these arrays. So it was like an investment. And then in the intestinal canal, we noticed there was an enzyme that was responding to microbes coming in, which was connected to tryptophan metabolism. And this enzyme is called TDO2. And this we never published, still haven't published this. But the fact that TDO2 responded to microbial signal immediately implied that there would be a link to regulation of monoamines. As you know, the tryptophan is the precursor to serotonin. And that led me to think there must be a possible link into monoamine regulation. So this was really the foundation of how I got into this.
Reflecting very much your metabolic and cell and biochemical background, but also then opening up a completely new window. Now, many people in Canada and somewhere else are, of course, looking into this, in particular in the behavior aspects of missing or present microbes, anxiety and stuff like this. Can you tell us a little bit where are you right now in this research and what is still missing? I think of an intriguing field which is the neurogenesis and the influence on the microbes on the new formation of the nerve cells, for example.
So with respect to behavioral phenotypes, I think there is sadly a tremendous amount of hype because people tend to cherry pick a strain or cherry pick a window that for sure they will see a phenotype that would enable them to write a story. So there's still a complete lack of mechanistic approaches. We have some interesting findings on fear, on electrophysiology, and this is about what I can talk about like this, but we clearly have some very, very interesting findings in amygdala.
Let me interrupt you and going back, and I really like that you put your finger on the hype in the field, which is really a danger. It's an emerging new field, there are good people in the field, but it's also there's correlative and papers which are not the quality we would like to see. The interest in this whole field comes because of neurodegenerative diseases in humans and the possibility that microbes and environmental derived factors could impact that, and that we can manipulate this type of environment. That's the base of, essentially. People use mouse models for disease models, also behavior models, autistic, so-called autism-like mice models. Do you believe that there is a future and there is information we can get out of that models when we look into human diseases?
You raise an important point, because one has to be very careful in terms of making interpretations when you are looking on the spectrum of human disease using non-human animals. And the phenomena or the facts still remain. A mouse is a mouse and a human is a human. So is there relevance to use animal models to understand symptoms that we in humans call disease? The answer is yes, it is. Because of the considerable amount of gene expression, patterning, overlapping, let's say, between rodents and humans. There are clearly regions in the brain which is very homologous, and then there are other regions which are very, very different. So to be very concrete in terms of one of the problems that you raised, which was the autism spectrum disorders, I would say the paper that came from Paul Patterson and Elaine Xiao captures elegantly what you can do using an animal model, where they identify a metabolite that has a structure homology to phenylcresol sulfate, which is found in high levels in children with autism spectrum disorders. It doesn't mean that it's causation, but it captures that there is something going on, because that 4-phenyl sulfate that they pull out from the microbe could mimic some of the behavioral changes that typically the behavior people would say, yes, this is an animal modification of change in social behavior that you may correlate to autism. So you can ask reductionistic questions once you know what to look for, but to look for causation, unless it's clearly a genetic drive, I think that's going to be a bit more complicated.
And very essential. Without causation, I mean, all these kind of comparative approaches will be very flat.
It is true, but if you take, on the other hand, neurodegenerative disease, and I can give you an analogy to the system that you have here. You are looking at contraction, and you have a phenomena that when the microbes come in, then you start to see a coordinated activity. Now, in my world, it is like if an environmental cue is involved in something when you start up, like in the developmental programming process, as we know the microbes definitely do contribute. Then you have a steady state period when you establish a level of homeostasis, but then comes when the host start to lose function. And now you start to see appearance of problems, and sometimes that is called neurodegenerative disease. So meaning that when you change the equilibrium between the microorganism and the host, this is where you have phenotypes, which is not uncommon in biology. So in this respect, the animal models can be very informative, because as you know, for example, Parkinson often comes with, oh, I had bowel problems for a couple of years, prior onsetting, meaning that the changes in the equilibrium, whatever that was, may have started in the bowel.
Right. a newly recruited colleague here at Kiel University, Professor Berg, told me once, Parkinson's starts 10 years before the neurological disorders in the gut, and nobody really knows the mechanistic basis for that. Yeah. Elaborating on that a little more, animal model systems, mouse, zebrafish, has also told us that, to our as developmental biologists, to a great surprise, that basic features of development, the development of the immune system, the development of the gut, as Karen Guillemin has shown in zebrafish, seem to depend, at least in a certain window, on microbial contributions. Even more fascinating seems to be reports which tell me that maybe even the development or the fine-tuning of the nervous system may have an interaction with the microbiome. Would you like to say a few words to the impact on the microbes on neurogenesis?
In terms of neurogenesis, I think, frankly speaking, the field is still not even open up. Is there any examples of, intrinsic, incorrect temporal and spatial development problems in a mouse not exposed to microbes? No, I don't think so. Does it mean that you can have structural changes? Yes, you can have. And I would guess gray and white matter is one. The myelination may be another one, because these are components which require energy. And if you're in shortage of energy, you have an issue there. And whatever the microbes are doing, they always do it for themselves. And foremost, it is about ensuring access to energy and saving energy by entering sort of a status where I consume less of energy. So that means that these dynamic properties, and as microbes came way before us, that is still a driving force, not because the microbes can think, but because they have the ability to supply the host with energy. And if the host then finds an ecological niche that gives an advantage for the surplus of energy, you can then see a beginning of selection of fitness. And in the case of the brain, that has to have been the case, because what is the brain? It's a big lump of fat. And that means that this would have cost an enormous amount of energy. And people have made the approximation that on a size basis, our brain would be about 700 grams, but in reality it's 1.3. So that means that there must be cues that compartmentalize and shunt energy at given time points in development. Is there an example of that? Yeah, there is. And there are some very elegant papers, both in mouse and in humans, that there is a window when you start to pump energy to expand the brain. Mm-hmm. And in children, in humans, this is about 4 to 7 years of age. And this must be driven by clearly hormonal changes. But one fascinating component in this may simply be a increased activity of the microbiome to provide cues and neuropeptides or guide neuropeptides to allow for NAD to be shunted.
This is, of course, particularly important then in this window of development in children if there is a disturbance or malnutrition, or so that may then really affect the morphological outcome and then also maybe the neurological outcome. Let's stay a little bit on this energy aspect or the metabolic aspect of the host microbiome. And we recently had Eran Elinav here, and you published just a few weeks ago or months ago, a very beautiful, another landmark review paper in Cell, which tells the community that there is, in essence, during our lifetime and within our own body, in all our different organs, there is no space without microbes and no time without microbial contributions. You talk about this interesting concept of inter-organ relationship, which is fascinating and in developmental biology not really understood. How is homeostasis maintained between the different organs? And how do you see the impact of the microbes in this more broader metabolic view?
It's still early days, but there are some interesting examples, and I will share one with you later in the seminar. But it is published, so I can talk partly about it. So microbes and microbiome-mediated pathways, either through the host nervous system or by secreting compounds, clearly can have impact on a number of key molecules that is connected to circadian rhythm and to changes in metabolic status. And one of these growth factors is the fibroblast growth factor 21, which we published essentially is not much on in a liver under conditions when it's not exposed to bacteria. Now, FGF21 has been, and there's a whole pleiotrophy of FGF hormones. They are basically involved in so many aspects of body function, and if they are subject to condition regulated by the microbes, you have a quite elegant way of having the microbiome to be able to modulate the signaling between organs. You have the lymphoid system, you have the nerves, and you have the blood system. So they are clearly there. But if you now add another level of modulation, which is secretory molecules, either from the microbes or induced by the microbes, then you have this level of cross-communication. And this is not well understood at the moment. If you ask Robert Perrimon, he will say this has to be done in flies because it's the ultimate system. If I talk to you, you may say, no, we should do this in Hydra. And I would agree that you can do it in elegance. It probably will be resolved in a more simple system. Taking this in more complex organisms like rodents, it's too complicated unless you do mathematical modeling. Then you may come up with it. But we stumble into the FGF21 as a pretty interesting molecule, and we also have found a link to a microbiome signaling activity that, again, I cannot really go into details, but the phenomena is there. So I think we have sort of unlocked something quite interesting, and that would be the tip of the iceberg. So to be then very provocative, since we know that microbes are confined to compartments, but they are not black and white. I mean, if you take bacon and egg, potatoes, and what you can have in a Western type of horrible breakfast, you see the microbes coming in. You see the endotoxin comes in. And then they are cleared. And this is just dynamic process. So that means that, of course, they will have an impact because they will provide signals to the vascular capillary to communicate to the organ next to it. And there's another example of dynamics which is poorly studied, and this is you have inflammation, but you can have swelling and sterile inflammation. And you can move then components in the tissue that come close to one another, which it's normally not there.
A human medical doctor, if he listens now, would immediately think, so what does it mean for my patients? And so maybe very briefly, a few sentences, how close is your work to in this type of translational aspects? I mean, are you collaborating with medical doctors? You are a medical doctor by yourself. There is still a lot of hype in the field if one thinks of the probiotics, etc. But how close is the research you're doing now on these metabolic interactions to going to the clinic or going to a patient?
To go into a patient, we are not there yet. That will take time. But I guess
which is an important message.
Even if we were to find some opening ends, I would not start with microbes. I would start with the components, the micronutrients, or the molecule that we identified, because this we can control. But the bigger consequence of this, and this is taking it from when the whole field said, let's clone and sequence the human genome. Then that was a big hype. Let's see if we now can track diseases. And that failed bitterly. But they gave us something very good. They gave us the sequencing tools. So with the sequencing tools, they open up so we can start to look into the microbiome community. How do they look like? And that has changed the field. The second point, and coming from your perception of the holobiont, when all these GWAS studies took place, they completely ignored the microbiome. And this is well captured in a Nature paper when they talk about genetics and obesity. And in a supplementary, there is a correction that the genetic contribution to obesity in the Western world is 1.47%. The rest is environment.
Yeah, too few people know that yet, and thank you very much for pointing that out. Yeah, you pointed out earlier that from the early days you studied and you got a degree in Sweden. Then you were recruited to the Karolinska. You are a professor at Karolinska, but from the early days you went to Singapore. Why Singapore? And now you are also a professor and an institute director in Singapore. Why Singapore?
Singapore, it was my former president who asked me to open up the dialogue, because this is 97, 98 when they decided to build the medical biology through the Ministry of Trade. And they were sort of called four musketeers that formed the Biopolis. And then they needed an advisory board in this Biopolis. And my president, because we hand out the Nobel Prize in Physiology and Medicine, he came in to sit in the advisory board, and he saw the opportunities. And then in a weekday, in a moment of my life, he said, Sven, would you like to see if this is worthwhile? That's how I got into Singapore. And then, as gradually I became fascinated, interested, change of culture, also where you are in your career. And that's something I never regret. But I mean, it's gone to the extent that I almost do not do anything more at KI because of the opportunity and the unmet need of medical science in Asia. There's a huge difference between Europe and Asia now. And it's going to be so for another 25, 30 years. So that makes it very, very interesting. And this address also your question about my relations to humans. Because in Singapore, the type of work you are doing would be much more difficult to do because they are so disease and clinically oriented. So what we have started to do is to transform some of our findings on how microbes influence locomotion and indirectly also behavior into human studies. So we have an ongoing project where we divide elderly, 65 and above, into physically active and sedentary. And then we have done pilot microbiome sequencing, and we have some interesting findings on this. We also now have preliminary data on middle-aged, and we have used students from the university as a control group. And then we see variations in the microbiome, and then we match phenotype information. Then we take individuals and transplant to the mice and look for phenotypes. And it looks quite promising. So the next level we are going now, and we just got the ethical approval, is that we have joined forces with UK Brain. And now we go for doing imaging of brain, liver, muscle to see the print. And then we have microbiome sequencing, and we're going to do selected metabonomics to build a picture of how do you look when you're healthy, sedentary, or physically active.
Mm-hmm. Fascinating. It's the holistic picture or the holobiont picture, essentially. So we are already far in the future. My final question goes to where do you see your research and the field in general in 5 years and then maybe in 10 years? What you expect we will have achieved and what is still a struggle?
I guess what has come out of the last couple of sort of more concentrated research conferences is a big need for mechanistic studies. And I am not sure that the research in the more complex organism will be the one that uncover the mechanism. I think it may come from the flies, it may come from your system in Hydra, because it has to be simplified. This is clearly an important window of research. So there's a lot of basic research to be made. The second point would be, and I was quite fascinated by a colleague of yours, because they built mathematical models on metabolic networks between the microbes. And to me, this is a quite interesting way forward, because you have to build models. And because the complexity of the microbes, how they communicate, it's impossible to take a reductionistic view. So you have to come up with some turkeys that build mathematical models. And from there, you come up with an hypothesis, and then you go back and test. This would be a second one. The third one comes actually from a friend of mine, which is Jeremy Nicholson and Elaine Holmes, basically looking into metabonomics in urine. Because the experience they've accumulated now tell you that if you want to get a print of the interaction between the host and the microbe, look at the metabolites in urine. And that is so simple.
Simple to get.
And it's so clean. And the power applying the machine learning. And we've seen what the machine learning can do, as, for example, in Eran's work. So that type of systems biology approach, I'm sure we're going to have some lucky guys who are going to stumble into some stupid molecule that is so important that, yes, you can capitalize on that. That's science, and I'm happy with that. And the final point is to bring in big pharma. We need big pharma to come in. And there is, of course, an issue here in terms of not being able to develop drugs that goes across continents, but rather to have targeted molecules for a region that have a enrichment of ethnic groups and have a food intake that would be tailor-made for some interventions.
Mm-hmm. Thank you very much, Sven. That was you covered a lot of terrain, and thank you for your time. Thank you for coming. We are looking forward to your seminar. Thank you.
Thank you.